Hello, I will talk about what the properties of adipose tissue-derived stem cells are generally and why the porcine adipose tissue-derived stem cells is chosen for our experiment. Firstly, according to one article whose title is the isolation and culture of porcine adipose tissue stem cells derived somatic stem cells, adipose tissue-derived stem cells have been described for a number of laboratory humans and animals. Improvement culture condition and cellular characteristic of adipose-derived stem cells have been identified. Adipose-derived stem cells can self-enable and differentiate into multiple tissue lineages. Further characterization of adipose tissue-derived stem cells in this manner could enhance isolation and purification of a population of mesenchymal stem cells from easily obtainable adipose tissue. This stem cells population from domestic animals, which make attractive model for transplantation studies, will be valuable for the evaluation of this efficacy in this tissue regeneration application in the future. These cells may also represent in a population more easily reprogramming during somatic cell nuclear transfer and thus expedite the development of transgenic animals from models and the development of valuable pharmaceutical proteins. And the other article for the porcine derived stem cell is porcine adipose tissue derived mesenchymal stem cells retain these proliferative characteristics, senescence, karyotypes, and plasticity after long-term preservation. According to this article, adipose tissue-derived stem cells, derived mesenchymal stem cells, decrease route cardiac functional deteriorations, even though a mechanism of the action or relevance of this finding to human condition is elusive. In this regard, the porcine model is the key translation step because it displays the anatomic and physiological features that are similar to those found in human heart. And in addition to this information, the characterization of porcine adipose tissue-derived stem cells are needed to positive or negative markers. Positive markers are CD29, CD19, CD44, CD140B, and CD105. And negative markers are CD312, 342, 452, and SLA-DR2. See you again.
Hello everyone, I am Ece and I will talk about the relationship between obesity and adipose-derived stem cells. I will summarize two articles about this subject. First article is related to obesity inhibits the osteogenic differentiation of human adipose-derived stem cells. Bone defects in craniomaxillofacial skeleton can occur as a result of congenital defects or acute injuries, secondary to trauma, surgery, infection, and cancer. Current options for treatment include the occlusive bone grafts and microvascular free plugs. However, drawbacks of these procedures included limited tissue donor infection is morbidity, unprotectable resorption rate, and high infection rates of both donor and recipient sites. Alternative strategies have been utilized, tissue engineering approach to supplement biodegradable scaffolds with the adipose stromal stem cells, and shown significant promise in treating craniomaxillofacial defects. Adipose-derived stem cells are self-neighboring multipotential stromal stem cells with the capacity to differentiate into osteoblasts. Adipose-derived stem cells segregate an abundance growth factor that assists in agnogenesis and bone regeneration. Animal experiments have shown the feasibility of healing critical size, clavicle, mandibular, femoral, and vertebral defects with adipose-derived stem cells seeded scaffold. Adipose-derived stem cells have also been shown to promote survival of endothelial cells and coordinate with local environment to form vascular networks to assist in healing. Exposure to chromatic inflammation may impact the stem cell function. Obesity is associated with low-grade chronic inflammation and is characterized by excess lipid accumulation and overproduction of inflammatory cytokines associated with adipocyte hyperplasia and hypertrophy. Studies have shown that adipose-derived stem cells isolated from these donor have loss of stemness marker and increased expression of inflammatory cytokines. Human adipose-derived stem cells isolated from lean and obese donors were characterized based on osteogenic differentiation in adherent cultures and on scaffolds. Critical size cervical defects were generated in nude mice and treated with scaffolds seeded with adipose-derived stem cells isolated from lean and obese donors. Both in vitro and in vivo analyses demonstrated the reduced osteogenic differentiation of adipose-derived stem cells from obese donors, which corresponded to change in gene expression profile. These findings indicated that obesity significantly reduces the osteogenic capacity of adipose-derived stem cells and that adipose-derived stem cells isolated from obese donors may be suboptimal as a cell source for tissue engineering application. The other review is related to obesity. The alternation in adipose-derived stem cells are particularly restricted by weight loss. The relationship between obesity and weight loss, according to the adipose-derived stem cell, is summarized in this article. The therapeutic potential of adipose-derived stem cell is reduced by various stress-inducing conditions that affect tissue homeostasis, such as aging, obesity, and diabetes. Previous works have provided evidence of negative effects of obesity on adipose-derived stem cells populations, but it's unclear whether this persists after a weight loss. This study evaluated whether weight loss can restore the attenuated properties found in adipose-derived stem cells derived from population with obesity. Obesity is a metabolic condition of excess calorie intake, leading to increased adiposity in peripheral tissues, including adipose tissues, muscle, and liver. Adipose-derived stem cells isolated from environment have impaired differentiation and migration properties. Excessive weight loss in patients with obesity has been shown to reverse the pre-inflammation state found in subcutaneous adipose tissues. Thank you so much.
Hello, my name is Büşra and I will talk about other applications of adipose-derived stem cells. Multipotence of adipose-derived stem cells makes them a subject for the other studies involving clinical applications, bone tissue engineering, and wound healing. Although clinical trials about these stem cells is in a few number, there is an article about standardization of isolating these cells which was published on Annals of Plastic Surgery in 1916. According to this article, these cells can be obtained easily from many different organisms involving human. Because white adipose tissue is the most abundant and accessible tissue in organisms. Multi-lineage differentiation and minimal donor site morbidity also make these cells valuable for regenerative medicine. To obtain more information about standardization of isolating these cells, you can check this article. Bone tissue engineering applications are thought to be a solution for extensive bone losses. In these situations, bone's healing capacity isn't enough for bone regeneration, so far bone marrow-derived mesenchymal stem cells are used in these applications However, there is an article about adipose-derived stem cells usage in bone tissue engineering. According to this article, strategies for regeneration of bone using porcine adult adipose-derived stem cells. These cells have similar features with bone marrow mesenchymal stem cells in terms of multilineage potential, immunophenotype, and transcriptome. High osteogenic capacity of these cells in vitro and in vivo increases healing of critical size defects. Scaffold and graft combinations of these cells can be used in anatomic defects, so these cells can be valuable in bone tissue engineering studies. Another usage area of these cells is thought to be about wound healing. According to another study about intradermal injection of adipose-derived stem cells and bone marrow stem cells to porcine models which have cutaneous wounds, healing is occurred in 14 days. It is demonstrated from this study that epidermal development is achieved due to regenerative potential, immunological properties of these cells. As you can see, adipose-derived stem cells may be used in different areas besides obesity. Thank you.
Thank you for listening us. Follow the other videos.